SARS-CoV-2 is the virus that causes COVID-19. The viral surface is studded with spike proteins, shown in teal, membrane proteins, shown in yellow, and envelope proteins, shown in red. In cross-section, the viral RNA genome, in orange, is wrapped around nucleocapsid protein, shown in gray. An individual can be infected with SARS-CoV-2 after inhaling airborne viral particles. These viruses can then travel into the airways, where they may encounter cells of the respiratory epithelium in the trachea and lungs. The spike protein on SARS-CoV-2 recognizes a specific protein called ACE2, which is shown here in purple on the cell surface. The spike protein, which is normally very dynamic and able to undergo opening, closing, and bending movements, is locked into an open position after binding to ACE2. Another protein on the cell surface, called TMPRSS2 and shown in orange, can then cut the spike protein in a specific location. These segments of the spike protein fall away, exposing a portion of the spike protein which was previously hidden. The spike protein is then able to undergo a series of dramatic conformational changes. During the first stage, the spike protein inserts itself into the membrane of the cell. In the second stage, segments of the spike protein zipper back on itself, forcing the membrane of the cell and the viral membrane to fuse. After fusion, the viral RNA is deposited into the host cell, where it will direct the cell to produce more virions. Once the nucleocapsid is inside the cell, ribosomes start translating the polyprotein precursor of the non-structural viral proteins. It is hypothesized that the progression of the ribosomes along the viral RNA results in the disassembly of the end proteins. The precise timing of polyprotein processing, however, is unclear. Soon after NSP3's protease domain is translated, it is thought to start cleaving NSPs 1, 2, and 3. NSPs 3, 4, and 6 are inserted into the ER membrane through an unknown mechanism, and NSP5 processes the remaining portion of the elongating polyprotein. Ribosomal frameshifting is a key event during early translation. It occurs at a slippery sequence located in the context of a pseudoknot structure just upstream of the first stop codon. If a ribosome melts the pseudoknot quickly, it progresses without frameshifting and then disassembles at the first stop codon. NSP5 then cleaves the polyprotein precursor. The pseudoknot refolds before it is encountered by a closely trailing ribosome. This time, the pause induced from the pseudoknot results in a -1 frame shift, allowing the ribosome to progress beyond the stop codon. In this case, NSPs 12 to 16 can be translated, and the polyprotein is processed by NSP5. In parallel, the ER undergoes a striking but poorly understood remodeling event. NSPs 3 and 4 interact to induce membrane pairing. This promotes membrane curvature. NSPs and the genomic RNA are trapped into double membrane vesicles, or DMVs, that form through unclear mechanisms. To propagate itself, the virus must replicate its genome and generate a large number of subgenomic RNAs in the DMVs. The viral genome is about 30 kilobases long, and its 3' end consists of genes encoding structural proteins. These genes are transcribed into a nested set of subgenomic RNAs. The first set of subgenomic RNAs are negative sense RNAs. These are later copied into positive sense RNAs that serve as messenger RNA templates for the translation of structural proteins. An interesting feature of the subgenomic RNAs is that they all share a copy of the leader sequence that is present at the 5' end of the genomic RNA. So how does this leader, or L sequence, get added to all the subgenomic RNAs? This process involves a series of sequences called transcription regulatory sequences, or TRSs. The TRSs are present at the junctions between each open reading frame of genes encoding structural and accessory proteins, as well as in the leader sequence at the 5' end of the genomic RNA. The core sequence of these TRSs is highly conserved, and the leader TRS is thought to remain in close proximity to a body TRS for base pairing. During transcription, when the RTC reaches a body TRS and copies its core sequence, it can either continue or switch to the 5' end of the genomic RNA, where the copied TRS matches with a leader TRS, leading to continuous transcription. In this way, all the subgenomic RNAs obtain the leader sequence at their 3' end. The RTC is a replication transcription complex made up of NSPs. However, its specific function is not yet well understood. In the next segment, we will show two hypotheses that are currently being tested. In DMVs, replication and transcription are carried out by the replication transcription complex. The core structure of the RTC consists of NSPs 7, 8, and 12. They assemble at the 3' end of a template genomic RNA and initiate transcription with the help of NSPs 10, 13, 14, and 16. It is hypothesized that transcription pauses when the RTC reaches transcription regulating sequences, or TRSs, shown here in yellow. Its helicase domain can then act on the double-stranded RNA, causing RTC backtracking and unwinding of the double-stranded RNA. The core sequence of the TRS is highly conserved. Therefore, when the copied TRS at the 3' end of a newly synthesized RNA matches with the TRS in the leader sequence at the 5' end of a template genomic RNA, transcription elongation continues. In this way, all the newly synthesized RNAs can obtain the leader sequence at their 3' end. In DMVs, replication and transcription are carried out by a replication transcription complex. The core structure of RTC consists of NSPs 7, 8, and 12. It is hypothesized that the genomic RNA first needs to be replicated in full length. NSP 13 binds to the positive RNA strand in transcription regulatory sequences, or TRSs, in order to keep an open bubble. Meanwhile, the RTC assembles at the 3' end of the positive genomic RNA and initiates transcription with the help of NSPs 10, 14, and 16. When the RTC reaches a TRS, it pauses and disassociates from the template RNA. Then, the RTC is recruited by the leader TRS at the 5' end of the positive genomic RNA. The core sequence of the TRS is highly conserved. Therefore, when the copied TRS at the 3' end of a newly synthesized RNA matches with the leader TRS, transcription elongation continues. In this way, all the newly synthesized RNAs can obtain the leader sequence at their 3' end.